getting down to the microscopic level of the heart itself. So prior to that, we were looking at the macroscopic level of the heart. We had gone through some of the anatomy of the heart. We were looking at some of the structures of the chambers, of the valves and things like that. When we were looking at the material from last week, what are either things that were confusing that we need to clarify, or things that seem to be really important that would be good to know for the test? Two things. What do you think? Blood flow from the heart to the lungs. So we said that that could be important for the lecture exam, where we have one of those flow types of questions or tracing questions where you're actually listing where that blood is going to and from. So for instance, from the heart to the lungs, it could start someplace. If you want, you could start, if we gave you a starting place from the right atrium through the right atrioventricular valve to the right ventricle, from the right ventricle through the pulmonary valve, up through the pulmonary trunk, split to the pulmonary arteries, and then you've reached the lungs, right? If you were doing the other side, from the lungs through the heart, then you'd be doing a tracing through the left side instead, okay? Yeah, so that's good. So it puts together some of the different anatomical features that we were talking about, and it has us imagining or envisioning how the blood is actually moving through, okay? What about another thing? Another thing that was important, or another thing that could be confusing?
The cardiac muscle can all be stimulated at once.
Yes. Yes, exactly. So that's one of the last things that we were talking about when we were talking about dead reindeer in Norway and that type of thing. We were saying that there's a coordinated effort of the cardiac muscle where it's working as what we call the functional syncytium, right? So it's working in sync, it's working as one big unit, even though it's thousands of cells. And that's because they have this connectedness, both anatomically and physiologically, where in these intercalated discs, these special cell-to-cell junctions that occur in cardiac muscle cells, there are gap junctions which allow the flow of charged particles, of ions, etc., to move directly from one cell to another. So we don't have to stimulate each cell in a chain or in a web. We can stimulate a few, and then it just moves through, much like conducting electricity, right? So if you have a direct connection, like if you have people that, you've seen those videos where somebody's like grabbing electrical wire at a fence or something like that, right? And they have all these people that are holding hands, it can directly go through them. And so same way, these cells are directly connected to one another, so an electrical event can directly pass from cell to cell, and it's almost instantaneous. Obviously, there's going to be some delay, but it's almost instantaneous, enough so that the atria contract all together, the walls of those atria contract and come in, and the walls of the ventricle, and back and forth and back and forth. Yeah, those are good points. Okay. So that leads us then to talking about those cells a little bit more in detail. So last time we had gone through and we had looked at some of their anatomy. We said that there are some similarities and some differences between cardiac muscle and skeletal muscle. And now what we're doing is we're saying, okay, there's that level of complexity that we have to remember that these are branched cells, that they have those intercalated discs and stuff like that, kind of that newer information. But now what we're saying is that, in fact, when we're talking about cardiac muscle cells, there are two different types of cardiac muscle cells even. So for the most part, the cardiac muscle cells that we saw in the diagram and that we were describing before are these ones that we see here that are contractile cells. So 99% of the cells that we find in the heart are contractile cells, making up that middle layer of the heart. What's the middle layer of the heart called? Myocardium, right? The thicker layer, particularly so on the left side, as we keep referencing. So those are contractile cardiac muscle cells, making up that myocardium for the most part. But in addition, there are some particularly specialized cardiac muscle cells that are called the autorhythmic cells. And we were saying these are kind of the elite, the 1%. These are the ones that are controlling. These are kind of like the managers that are controlling all of the other ones. So autorhythmic cells are, in fact, cardiac muscle cells. Their shape is a little bit different, as you would imagine. They're not quite, they don't quite look like with the striation patterns like we see with the contractile cells. But their job, as the name indicates, is to kind of on their own in a regular way generate a rhythm, generate that heart rhythm. So they are kind of the pace setters, if you will, of the heart. They are the ones saying to the contractile cells, okay, contract now, 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 now. Faster or slower depending on the needs of the body at the time. If you are, we have a Red Mountain Running Club here on campus, and if you decide that one day you're going to come out with us and go running around campus, then you can expect that during that time when you're running, when there are increased demands around the body tissues, that your heart rate is going to increase, right? So it has to get feedback through homeostatic mechanisms to say, okay, there's more demand, so we need to increase our output, and so it'll increase. Now that's going to be a feedback mechanism from certainly the central nervous system, right, getting the sensory information about oxygen debt and things like that. There are going to be direct neural pathways down to the heart to influence it, to say, okay, you guys down there in the heart, you need to speed up. That's a bit like if you want to think about it in terms of the workplace, that's a bit like your regional manager or something like that. This is somebody who oversees from a distance, and then it influences what's going on a local level, right? So those types of like sympathetic and parasympathetic impulses coming down definitely control the heart activity. But once it gets to the heart, then these local managers, these autorhythmic cells, are the ones that then route that message out to the worker cells, which are the contractile cells. Okay? Yeah?
So the autorhythmic cells, are they throughout the heart, or are they mostly found in the SA node?
Yeah, that's a very good question. So the autorhythmic cells, like we said, are going to be the ones that initiate the rhythm, the action potentials. We're going to talk about their membrane potentials and things like that and how it works. But these, as the name indicates, pacemaker cells, pacemaker potentials, they are going to be found along what's called the conduction system of the heart. So the pacemaker of the heart is called the SA node, right? The sinoatrial node. It exists on the wall between those different atria. That is a big knot or a big core of these autorhythmic cells. They're all in that one area. But from there, we'll find that there are other parts of the conduction system of the heart. That's where it starts. And then there are kind of radiating patterns of these autorhythmic cells which take that signal from, for instance, the right atrium to the left atrium, and then from the atria down to the ventricles so that they get the message that it's their turn to contract. So the autorhythmic cells, there. We'll come back to those other ones, don't worry. The autorhythmic cells are found where you see these yellow lines here. So in the upper corner right there, this is the sinoatrial node. That is the pacemaker of the heart. So that's the first major concentration of our autorhythmic cells. But you can see, right, radiating lines of them heading out to here. This is the second point of our conduction system, a second knot, if you will, of autorhythmic cells. This is called the AV node. Then we're going to see a pathway that leads down to the ventricles through what's called the AV bundle, or the bundle of His. It actually occurs in girls too, so we call it the bundle of hers in that case. So the AV bundle goes down between the atria and the ventricles, and then you'll notice that it splits. So these are bundle branches so that that signal can reach all of the myocardial cells or the contractile cells on both sides of the ventricles, the left ventricle and the right ventricle. Okay? So point being is that most of the myocardium is going to be our contractile cells, but the autorhythmic cells, which are kind of the managerial cells, exist within this conduction system of the heart. And when we look at this figure, before we were talking about it, before you were reading about it, you would look at that and you're like, oh, I see, those are neurons, right? So that just looks like nerves and neurons like we see from when we were talking about nervous system. But in fact, these are specialized cardiac muscle cells that are doing the job that a typical neuron would do. It's routing or communicating with cells throughout the heart. Okay? All right. So this is where our different autorhythmic cells, the 1%, can be found. we say that they have a different job to do. They are working to conduct and coordinate the activities of the heart, and they do so in a specialized way. So these ones, like we said, the 1%, they have an unstable resting potential. Now we're starting to get to those terms that you were scared of before when we were talking about nervous system and muscular system, right? If you remember, when we were talking about the nervous system and the muscular system before, we talked about these things called action potentials, right? So if you were drawing that out, did any of you have to like draw that out on exams or anything like that? Maybe some people. Sometimes on my exams you have to do stuff like that. But if you were drawing the graph, right, typical XY graph, right? So on the x-axis here, we had time. On the y-axis here, you had voltage in units of millivolts, okay? So if we were drawing an action potential in terms of like a regular neuron, what we had was something that kind of looked like this. And it came up a little bit, then there was this big spike up and down like this, and then it went back to being flat, right? So this whole thing is one action potential. And what's happening here is in a neuron, if it's not doing anything, we said at about -70 millivolts, we have what's called a resting potential. So a regular neuron, if it's not sending signals, it's just sitting there resting, and its membrane potential, comparing its charge inside the cell compared to outside the cell, it's more negative inside the cell at about -70 millivolts. Reason being, right, we have this distribution of charges of two different ions that are important. What are those two ions that we were talking about before? Sodium and potassium ions, right? So we had a whole bunch of sodium ions piled up outside the cell. We had a whole bunch of potassium ions piled up inside the cell. And if we opened up channels, because there was that gradient, they would rush one way or the other, right? Open up sodium channels, and sodium would rush into the cell. Open up potassium channels, and potassium would go down their concentration gradient and rush out of the cell. That's what we're seeing here, right? This part of the curve was called depolarization, right? So this is depolarization, which was due to the movement of sodium going into the cell. So at this point, positively charged sodium ions were actually going into the cell, and that was making it less and less and less negative, right? So think about your lives. If lots of good things are happening to you and you were in a bad mood before, more and more positive things coming in make you less and less negative, right? So this is depolarization because sodium's coming in. This part is called repolarization because potassium ions, positively charged potassium ions, are leaving the cell instead. So again, the analogy, right? If you are having all these positive things leave your life or whatever, then it makes you more and more negative. That's what we're doing. We're coming back down towards this resting potential again, okay? So very fast, depolarization, repolarization, back to resting potential. That's what we saw with action potentials. We would repeat these waves down the axon until we got to the end of the axon, and then that would trigger a release of neurotransmitters, which would then, like a baton in a relay race, get that signal to the next cell. The next cell would generate these, and then the signal would continue. Okay? If that doesn't seem familiar to you, you may want to review it a little bit. But that's a review of what we saw before. So in excitable cells, they utilize the movement of charged particles like this in order to relay signals or generate muscle contraction, like in skeletal muscle, etc. All right, so that's the basis of what we know so far. So how are these specialized cells, which act like neurons, how are they relaying their signal? Does it look exactly like that? Is it something different, etc.? So that's what it looks like in an autorhythmic cardiac muscle cell. So in some ways, looks the same as what we were just talking about here, as in a neuron, but in other ways, it looks different. So we definitely see the peaks and the valleys, the depolarization and the repolarization, but what looks different in this curve compared to the one on the board? So definitely we're seeing calcium ions influencing this. We don't see that here. And what else? There's no resting potential, right? There's no flat area of that curve for autorhythmic cells. That's important because if your heart is resting or flatlining, that's a bad thing, right? We do not want the heart to get tired and have to take a rest because everything else suffers accordingly. So there's no resting potential in these autorhythmic cells. They are constantly, consistently depolarizing and repolarizing. When they get done repolarizing, okay, we're starting to depolarize again, okay? Always going from one to the other. Now, the rate of this can increase and decrease. We can send more signals or less signals, but it's always going to be going from one to the other. So when we look at it, we have no resting potential, okay? The other thing that we said that was different is that we've got calcium channels here. So rather than seeing sodium in charge of our depolarization, we see calcium is responsible for our depolarization in these cells. It's still a positively charged ion, so when it goes into the cell, it depolarizes the cell, but calcium is responsible for depolarization here. Repolarization is the same. Repolarization, we lose potassium ions out of the cell, and it brings it back down again, okay? Technically, there's kind of two different types of calcium channels. There's some slow ones that are here. There's some fast ones that go here. But it's calcium that's responsible for depolarization there. And these calcium channels that we see here, okay, there are some sodium channels that are involved as well, but it's the calcium channels that do the fast depolarization, the big spike that we refer to as an action potential, okay? So that's what's going on in terms of our autorhythmic cells. No resting potential, constantly depolarizing and repolarizing and sending these signals down the conduction system of the heart. Okay, so that's part of, like we said, the conduction system of the heart that's located inside of the heart itself. They're distributing and initiating these impulses so that we can tell the rest of the cardiac muscles what to do. So we said these are just the textural parts that go through what I was talking about, right? So we have some of these slow sodium channels to begin with that we see here that leads to the calcium channels opening up. So it's mostly calcium channels that are depolarizing the cell. Repolarizing the cell is due to potassium channel and potassium leaving the cell like we saw before. Okay? Like we saw in the diagram, there's a bunch of different parts where these autorhythmic cells are found. This is just talking about those different parts like we talked about before. The only thing that I didn't mention when we were looking at the graph, or I'm sorry, the figure coming up, was this thing that's called the AV nodal delay. So this is what we were saying. SA node is the main beginning point, the pacemaker of the heart. Then there's the AV node, then the bundle branches, the AV bundle, the bundle branches, and then these ones that wrap around here are called Purkinje fibers, okay? But we said last time when we're contracting the heart, we have the atria contract first and then the ventricles. Now we know that electrical signals in the body travel very quickly, right? So you can stimulate the muscles in the bottom of your foot almost instantaneously. You can't tell the difference. You're sending a signal, right? I'm sending a signal right now from my brain down to my foot. I mean, it goes so fast, I can't even say it fast enough for it to be happening. And so when we look at this, it's also very fast, but we don't want this signal to spread throughout the heart immediately. Every part of the heart would just contract at the same time, and we can't have that, okay? We need the atria to contract first to push the blood down to the ventricles. The ventricles have to be relaxed in order to receive it. Then the ventricles are going to contract while the atria are refilling again. So we need this alternating contraction and relaxation. So at this point that we see right here, there is part of the heart which is connective tissue. It's called a fibrous skeleton of the heart. It's not bone, but they call it a skeleton because it serves as an anchoring point for a lot of those valves. It serves as an attachment point for those cardiac muscle bundles like we were seeing before. And that connective tissue is not very conductive, right? So muscle tissue and nervous tissue is very conductive. Signals spread through it very quickly. But the connective tissue going across right there between the atria and the ventricles does not conduct electricity very well. So when that signal is coming from the atria down to the ventricles, it actually slows down as it's going through that wall. And so that's good because it gives the atria a chance to respond and contract first. And then there's a little bit of a delay before the signal gets to the ventricles, which means the ventricles are going to get the signal later and they contract afterwards. So atria, ventricles, atria, ventricles. So that's that AV node delay, is that as that signal is moving down, it passes through that dense regular connective tissue, slows it down just enough so that the ventricles are still relaxed when the atria are contracting, and then it reverses. Okay? Okay. So if we look at a comparison of these action potentials in different areas of the heart, again, we see time on the x-axis. This is going to be millivolts on the y-axis here. We're comparing action potentials or potentials in different areas. SA node, like we saw there, in the SA node right there, we are going to see the increase and the decrease, and it's going to keep continuing. That will continue throughout. Like we said, it goes back and forth. But look at the shapes of these other ones here. When we compare the muscle, the contractile muscle, either in the atria or the ventricles, it again has this weird looking shape to it. It's not exactly what we've seen before in terms of an action potential. Okay? So it has definitely a depolarization that occurs very quickly. It has a repolarization that occurs very quickly, but it has this big flat part that's called a plateau. Okay? So with that, the way that that works, either in the atria or the ventricles, is these parts are the same. Sodium going in, good. Potassium coming out, good, same. Okay? This part right here, this flat part, the plateau, is again due to calcium. Okay? So calcium in the heart has a direct role in terms of the activities and membrane potentials of these cells. Okay? Back before with skeletal muscle, we saw calcium had a role as well as a second messenger coming down and allowing the myosin and the actin to interact. But in this case, it's actually changing the membrane potential and influencing the action potential itself. So if we have calcium coming into the cell at this point, what it does is it keeps the inside of the cell more positive for a long period of time. So it keeps it in a depolarized state for a long period of time, and then when those calcium channels close, then the potassium brings it back down again. Okay? The importance of that plateau, notice that it can be like 200 or 300 milliseconds. Now we're saying milliseconds, so that's really short, but 200 to 300, this whole thing, this action potential here, is like 6. Now we're seeing an action potential that is like 300 or more instead. So the importance of this delay that we see here, the plateau, is that when we are contracting the atria or the ventricles, we don't want a little twitch like we see in skeletal muscle. Skeletal muscle, it's okay to have a twitch, and then we relax it again. But in this case, with the atria or the ventricles, we want a very forceful squeeze of the chambers, right? Imagine, if you will, you have a sponge that's full of fluid, and you want to get the fluid out. If you just do wink like that, right, you're going to get a little bit of fluid coming out, and a lot's going to be stuck in there. Same thing would happen with the heart. If we just do a little twitch, then you're going to get a little bit of fluid exiting, and there's going to be a lot that remains, right? It's not going to be a very efficient pump. So we want to make sure that all of those atrial cells or all those ventricular cells squeeze and give a chance for all of that blood, or most all of that blood, to exit before we then go into a relaxation phase and allow it to fill again. So that plateau that we see there with the contractile cells due to calcium is very important in allowing the blood to fully exit that chamber and squeeze it out before we relax it and allow it to fill again. Okay? So it's very important that we have that plateau there, an extended period where we're going to squeeze. It's important that it's a regular type of squeezing and then relaxing, squeezing and then relaxing. If you don't have a regular beat, then that whole pump, in terms of filling and emptying, it gets thrown off. So if you have, for instance, an arrhythmia where that pattern is disrupted, too fast or too slow or irregular, that's arrhythmia, right? A just means without. So without rhythm, that is a big problem in terms of an efficient pump. Fibrillation is where you have kind of spasming. Now, if it's atrial fibrillation, it's not as bad, right? One of the things we said is, like, most of the blood in the atria naturally just dumps down into the ventricles when they're relaxed, right? So it's only the last 20 or 30% that we actually have to shove down. And so if you have atria that are kind of spazzing out, most of the blood can still get down to the ventricles with no problem. But if you have ventricles that are spazzing out, that's bad because that's where we're really pushing the blood to the lungs and to the body. And so we need that to be efficiently moved. Okay, so what we find, don't worry, like over here where it shows permeability and stuff like that, it's pretty. Don't worry about having to reproduce that on an exam or anything like that. But it just shows you how sodium permeability, that means movement of sodium, is causing that depolarization like we said. How calcium permeability goes up and maintains that plateau like we mentioned. And then the potassium opening up, potassium channels opening up later produces the repolarization that we see here. Okay, that's all that one on the right says there. But this is a change in the membrane potential for a contractile muscle cell. And it has a long period where it stays depolarized. We said that's important in terms of getting that blood moving. But then it also has a long what's called refractory period. So a refractory period is a time when that cell is not going to be able to respond the same. So in a muscle cell, the refractory period is pretty slow. I mean, pretty fast, like we can get it to generate another twitch very quickly. Same thing as in a neuron, okay? But in these cells, the refractory period is like, oh, I got to recover, right? I'm tired from that last whole effort. I got to recover a bit. So the refractory period that we're talking here in cardiac muscle cells is also really, really long. You can see it's almost as long as that contraction period. And that's important because we said that when contraction's happening, that's when we're pushing the blood out. Emptying is a very important stage. But equally as important is relaxing and filling. It's reloading for the very next cardiac cycle. It's not a one shot like, oh, great, I pushed the blood out. Now I'm good to go for like an hour or so, right? Right? Less than a second later, you need to be pushing blood out again. So you have this continuous pushing of blood throughout the system. So the refractory period makes sure that we can't stimulate that muscle again and get it to be doing this twitching type of thing. We stimulate it, it does a good contraction, and now it says, I'm not hearing anything else for a while. I got to rest, I got to rejuvenate. I'm in this refractory period. I'm going to go into this relaxation. I'm going to spend some time filling. Then we're going to get back and I'm going to be able to do this again. Okay? So it has a long contraction period and it has a long refractory period to make sure we have this very regular pattern of filling and emptying. Okay? Excuse me. So this one that we have here, this slide that we have here, just goes through what I was talking about. So it is important information, but it's nothing new there, right? Depolarization due to sodium channels, plateau or flattened depolarization due to calcium channels, repolarization due to potassium channels, and then a long refractory period so that the heart can fill again. Okay? Okay. So all of this slide as well is just talking about the very same thing that I was going through on the graph. So I'm not going to go through it in terms of each individual word there, but it's the same information that we were just talking about. So the big picture there is you should know what's going on down on a cellular level. You should know the difference between contractile cardiac cells and autorhythmic cells, where they're found, relatively how much do we have, are they mostly autorhythmic, are they mostly cardiac, contractile cells, and you should be able to describe or draw what's going on in the cell when it's doing its job. So this one we said up here, that's for like a neuron or skeletal muscle cell, etc. You want to be able to draw those other two graphs, like in the autorhythmic cell, the ones that goes back and forth and back and forth and back and forth. Be able to know what causes depolarization and repolarization, etc. Same thing for contractile cells. That kind of, if you do cursive, you don't do cursive anymore, maybe for your signature or something like that. But a cursive-shaped R, lowercase r, goes up like that. It has a flat period, and then it comes down. That's the type of curve you got to know for contractile cells. You got to know sodium, calcium, potassium. That's the plateau period. Why is that important, etc. Okay? I know that when we're talking about ions and chemicals and what's going on inside of cells, it's a difficult thing because it's not as big and obvious as having a model and pointing out parts of it. But that is what gives rise to the heart being able to do its job of pumping blood and sending it around the body, etc. That's what it comes down to, is these two types of cells doing their job and having these membrane potentials change like that. Okay? So it is pretty important. So associated then, do you have questions on membrane potentials and cardiac and those two types of cells? That may be one of those things when we meet next time on Tuesday, you're like, okay, that's the part I think it's pretty important. Maybe we can go through that again? Probably. Right now, it's just kind of, all sinking in. So make sure you look at those figures in the textbook, go through them yourself so you can understand them, etc. Okay? Okay. So what we were talking about there was the electrochemical activity that's going on in individual cells down in the heart, whether they be an autorhythmic cell or a contractile cell. Now, that shows us that there is a lot of physical activity in the heart, but a lot of electrical activity in the heart, movement of charged particles this way and that way, etc. So what we find is that one of the different heart tests that can be done and that we are going to do, not next week, but the following week in lab, is you can actually measure the electrical activity of the heart using an EKG or an ECG. Okay? Now, an EKG or an ECG, once upon a time when they developed this, developed by Germans, and they spell cardio with a K. So that's why they call it an EKG. But we, here in Merca, call it electrocardiogram because we spell cardio with a C. So you may still hear it if you work right in the hospital or something like that. They may still refer to it as an EKG, but K is the German spelling of it instead. All right, so the deal here is that with an ECG, what you're measuring is not what's going on in an individual cell, but kind of a summary of all the electrical activity that's going on in the heart. When the atria contract and when the ventricles contract, when they're getting that signal, when they're kind of repolarizing and relaxing again. So it's a summary of all that. And luckily, our body tissues are very conductive as well, right? The fluids that are throughout, extracellular fluid and blood and even our muscle tissue, etc., it's conductive. And so those electrical activities that are going on here actually spread throughout the body, and you can measure them on the surface of the body. So if you're having an EKG done as part of your physical or because they want to see what's going on as you get admitted to the hospital, luckily they don't have to put electrodes down on the surface of the heart and try to sense it that way, right? They can put electrodes on the surface of your body, and you can still get a measurement of what's going on in the heart. Now, that being said, any other movement that you have going on, so if you start moving your limbs or you're doing big breaths or something like that, those are muscles that are also moving charged particles. So you start doing that, and it starts throwing off the EKG. So when you're doing one of these, the patient has to be almost immobile so that you can get basically the activity of the heart coming through itself. Okay? So you should see a very regular pattern of repeating three waves like you see up there. Now you may see, some sort of other problems here where you're seeing extra waves, or in this case, we don't see a P wave, the first wave that we should be seeing of the three. In this case, right, we're seeing big arrhythmia there. We're not seeing a regular pattern of atria and ventricle activities. But when we look at it, and we zoom up, if you will, on one of these series of three repeating waves, what we see is there's a little one to begin with. It's called a P wave. There's a large one in the middle that's called a Qrs wave complex. And then there's another little one that's called a T wave. And it's those three waves that repeat again and again and again in an ECG. Each time we go through a cardiac cycle where the atria contract and then the ventricles contract, we're going to see that series of three waves as electrical activity, okay? Or at least we should. So what's happening here is the signal, like we said, is spreading from the atria to the ventricle through the conduction system, and then all those contractile cells are responding. Sodium moving in, potassium moving out, all of that. So what's happening here is the very first bump that we see right here is due to atrial depolarization. So the P wave is where the atria are first getting that signal that it's their turn to contract, okay? So that gives us a little bump right there. And again, remember, this is showing a sum of the electrical activity. And it's showing it for all of the cells of the atria. We're not looking at one cell like we were before when we were looking at this, or the R-shaped, if you will, contractile cells. This is a sum of all the activity. Atria are smaller than the ventricles, so to a certain extent, we'll see a smaller bump, and this is our depolarization where they're first getting the signal. This part right here, the QRS wave, two things are going on. The atria are now going into repolarization when they're going to relax, and now the ventricles are getting their turn, their stimulation to start contracting. So this QRS wave here is especially big because there's two bits of electrical activity that are going on. The atria are repolarizing, and now the ventricles are depolarizing. So they add up to a very large wave that we call the QRS wave. Okay? The last thing that we see here, the T wave, is when the ventricles are now, they've done their signal to contract, now they're getting their signal, they're repolarizing, they're going to go into the relaxation or filling phase. So the T wave that we see there is ventricular repolarization. So it kind of looks like this, right? The SA node gets the signal to begin with, it spreads to the rest of the atria, and we can see when those muscles are starting to contract or starting to get their signal, that's our P wave that we see down there, okay? That signal is going to pass then from the atria to the ventricles. Now the atria are going to be going into their relaxation phase, and the ventricles are starting to get stimulated. So now we get this big QRS wave here as the atria are relaxing, and now the ventricles are getting their stimulus to contract. Then finally, this last one that we see here, the T wave, is when the ventricles are going to go through repolarization and going to their filling stage. So the waves, the three waves that we see there, have to do with the signals arriving in all those contractile cells, either in the atria, depolarizing and then repolarizing, and then the ventricles depolarizing and repolarizing. The actual contraction or filling occurs kind of in those gaps, those flat areas that we see of the curve instead. Okay? So the ECG waves, you should be able to identify those three waves, be able to label them P, Q, R, S, and T, and then describe what's going on during each. Atrial depolarization, then atrial repolarization, ventricular depolarization, then ventricular repolarization. Okay? If a wave is missing, if a wave is too big, if this rhythm is off, it gives us an idea. Maybe there's damage to the heart. Maybe we've had cells die along the atria or the ventricles or something like that. Maybe there's been some sort of a heart attack where cardiac muscle cells have died off, or they're being restricted in terms of blood flow or something. Okay? Okay, so changing in those patterns of the ECG then can give us an idea that there's some sort of issue. When we look at the QRS wave, like that R wave, the big peak, if you have enlarged ventricles for whatever reason, then that peak is going to be even bigger because there's more cells there contributing to that signal. If you look at this ST segment, that's again between the S wave here and the T wave here, that's where the ventricles are supposed to be contracting. If you have cardiac ischemia, where basically you have a reduced blood flow in that area and a reduced responsiveness, then you're going to see an area of that wave that doesn't appear quite right. Okay? So ECG is one of the ways that we assess the activity of the heart. Is it operating normally or not? All right. Now, we talked about electrical activity occurring in the heart itself with this conduction system, but we also said that there is overall control coming from the nervous system before. So when we're talking about unconscious level control of organs, what branch of the nervous system is that? Everything's being controlled automatically, and so it's the autonomic nervous system, right? So autonomic nervous system has two different branches to it. It has sympathetic and it has parasympathetic. What was the phrase that goes with sympathetic? Fight or flight, right? It prepares the body for kind of a stressful situation, whether it's physiologically stressful, emotionally stressful, whatever, right? It prepares the body for a stressful situation. Parasympathetic, the phrase that goes with that is rest and digest. So it prepares the body for kind of a maintenance, low-level type of physiological situation instead. So they're opposite usually in terms of when sympathetic does something, parasympathetic usually does the opposite. It's a bit like gas pedal and the brake. And for most systems, sympathetic is going to speed it up and make it more intense, and parasympathetic is going to slow it down. It's not always the case, though. So for instance, parasympathetic, rest and digest, digestive system with parasympathetic stimulation, that's going to increase the activity of the stomach and the intestine, etc. It's their time to process that food as you're laying on the couch in the food coma, right? So when we talk about digestive system later on, in any of these systems, we talk about how the system works by itself, and then we look at a bigger picture. How does the nervous system work to control and coordinate it with the other systems of the body? So that's what we're doing here. So in this case, with the heart, we have sympathetic and parasympathetic control going to it. So we have sympathetic neurons that go down to it. You can see how they control and coordinate with those local managers, right, the pacemakers of the heart. And there's also parasympathetic stimulation going down too. Vagus nerve is cranial nerve number 10, right? It's one of the few cranial nerves that's not located up in the cranium necessarily. It has branches that go there, but it also goes down into the thoracic cavity. So if we need to increase or decrease the heart rate and the strength of contraction, this is the way that we can do it. A little bit more gas pedal, a little bit more brake. We can finally control the actual rate of the heart, okay? Okay, so in terms of heart activities and assessing the heart, etc., we said that ECG is one of the ways that we can do it, and we'll be doing that in 2 weeks. The other way that we can do it in terms of normal heart function is we can listen to what's going on. So we can do auscultation. So if you go and do a physical, oftentimes they will use that stethoscope and they will listen to certain things, right? They'll listen to your heart, they'll listen to your lungs. They often listen to your digestive system as well to make sure that things are passing through. Those kind of gurgling sounds are actually called borborygmi. We'll talk about that with digestive system. But for the heart, we're listening to sounds in the heart, the normal bump, 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 bump. So when during the physical, the doctor is placing the stethoscope, it's not usually just like right at your sternum and listening to the heart. You can certainly hear that, right? You can hear that on somebody else just with your normal ear, but he moves it around, he or she, right? So it's not that he or she is lost. They're like, oh, I can't hear it there. I'm going to try over here, try over there. By moving the stethoscope to distinct points, usually between the ribs, right? So that you can get to the soft tissue there, you can highlight more of the sounds or the activities coming from the 4 different valves that we have inside. So it's the valves that are making these noises that we call the lubbed up sound, right? So these sounds are associated with the valves that are closing. Now remember when we were talking about the valves closing, we were saying that if we imagine this as being one of the rooms, this is going to be like the atrium where you are here. When the atria contract, imagine the walls coming in. That's going to decrease the volume and it's going to increase the pressure. Pressure for you guys is like, it's too crowded in here, I'm bumping into people, I want to exit, right? So what you're going to do is you're going to go to the nearest door and you're going to exert a force and push on the door and you're going to go into the next room or go into the hallway in this case, where it's actually a little bit cooler, so I'm going to hang out here for a little bit. Anyway, so you exert a pressure on that valve, you open the door, and then you're in the next chamber. Now, if you wanted to come back, right, this blood can only push, it can't pull, right? So if the blood comes back, like when the ventricle, imagine that hallway starts contracting and getting smaller, and you start panicking out there, you might try to get out of the hallway, you try a door. If you push on the doors to come back in here, all it's going to do is close the door faster and more forcefully. So when the ventricles contract, they push on the valves. What happens is it catches in those umbrella-shaped cusps, and it brings that valve closed like that. Now this is a hard door, so when you move the door into the door jamb, it makes a noise. In truth, the noise associated with the valves closing comes from the blood rushing up against those cusps, and when it closes it, it goes from a nice smooth laminar flow to a kind of an angry, turbulent flow instead, right? It's like at the door going, oh, I want to go out here, and I can't, and so it starts swirling around like at a river or a waterfall. And you know, if you're at like the waterfalls or something like that, there's a lot of turbulence, it's really loud, right? So that's what's making the noise of the bump, 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 bump. It's this blood closing the valve and then doing this turbulent mixing next to it before it gives up and then exits and goes out the other valve instead. Okay? So to be sure, the heart sounds that we hear are associated with the valves closing, but it has to do with the blood doing this turbulent mixing next to the valve. It's not like the valves themselves are slamming closed. It's that turbulent movement of blood next to the valve which actually makes the noise that you can hear. So the first noise comes from the AV valves closing, and the second noise comes from the semilunar valves closing instead, right? Atrial ventricles, etc. So that's where those noises come from. We will be doing that as well in lab when we do our physiological lab week after next. We're going to be listening to those. But the first sound comes from the AV valves closing. Second sound comes from the semilunar valves closing. So we've noticed, as we've been talking about the chambers contracting and moving the blood and stuff like that, that we're starting to see these terms here. So these are terms that are important for you, systole and diastole. When you look at those, it's like looks like systole and diastole, but it's diastole. You pronounce the E. It should have a little accent aigu, technically. But systole is where we have contraction of the heart muscle. Diastole is relaxation of it. Remember, we said that atria and ventricles, they contract and relax at different times. So there is atrial systole, and then ventricular systole, and then atrial diastole, etc. It alternates. So it's not just heart systole. That means the whole heart would be locking up, right? It's atrial systole first, then the ventricular systole. But those are the terms that we use. So when we look at it, when we're talking about the heart and its activities, we're constantly assessing in allied health sciences how well the heart is working. So we're looking at the ECG, we're listening to those valves. Remember, if those valves don't close tightly, you can get what we were talking about as a heart murmur, right? Where some of the blood is leaking back into the chamber where it shouldn't. So rather than having that nice staccato, bump, 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 it's bump, shh, because some of it is leaking back rather than driving that turbulent flow instead. It's able to sneak back instead. So we're looking at assessing whether the heart is doing its job efficiently. One of the other things that we look at in terms of the heart activity is something that is called cardiac output. And this is coming up on a couple slides as well. So I'm just mentioning it here because as a pump, that's what we want to look at. Is this pump working? How much fluid is it putting out per unit time? How much pressure is it generating per unit time, right? Those are important aspects of a pump, like a pool pump, right? It has to have enough pressure, push it around into the pool, back and forth. And so when we look at it, what we find is that we can even assess how much blood is being put out and how much is left inside of these different chambers, okay? So for instance, this thing that you see here, it's not critical. We're not going to be going through and assessing it in lab or anything like that. But there is something that's called end-diastolic volume. So diastolic refers to what? Contraction or relaxation? Relaxation, right? So end-diastolic volume is looking at the volume of blood in each ventricle at the end of relaxation. So relaxation is when that chamber is filling. So that gives us an idea about how much blood has it reloaded with. That's how much it's potentially going to eject next time. So how much should we fill it up? Like if you're filling buckets like in a relay race, it makes a difference how much you have per bucket, right? If you just fill it for 2 seconds and then run over and dump it over here, you're not transferring a lot of fluid. If you let it fill a lot, it takes a little bit more time, but now you have a full bucket that you're going to empty out over here. So end-diastolic volume is how much these ventricles have had a chance to fill and reload before we're ejecting it again. So that's something that they will kind of assess is end-diastolic volume. There's also this thing called end-systolic volume. So this is important because when those ventricles get their signal to contract, we want to try to push all of that fluid out as much as we can. Again, like squeezing the sponge, if we have a full sponge with a lot of fluid and we just go, whee, then you only get a little bit of fluid and there's a whole bunch of fluid left in that ventricle. So we can see if that ventricle has had a chance to really fill up, what's that volume, and when it contracts, how much is left in that chamber. Did it eject almost all of it, or is there some that's left there? If you start having a heart condition where you get an enlarged heart or something like that and the muscle starts getting weaker, the volume may be just fine when you're loading it up, but it doesn't have the strength to fully eject all of that. So you're having a residual amount that's sitting in there. That's not a very efficient pump in terms of circulating blood, okay? Okay, so we can look at the end, like we said, systolic or diastolic volume. There's also this interesting stage here that nobody thinks about a lot, which is called the isovolumetric contraction phase. And you're like, whoa, that's really long in terms of words, right? Iso means what? So like isotonic, isosceles triangle, things like that. You're like, I so don't remember that. That's what it means to me. That's all, right? Iso means the same. So isotonic means it has the same concentration inside and outside the cell. Isosceles triangle, the sides and the angles are the same, etc. So isovolumetric contraction means that the volume of these ventricles that are all loaded up, it's not changing yet. They're basically starting to contract, but it's not actually changing yet. What it's starting to do is generate pressure. And so the volume of blood isn't moving anywhere yet. We're starting to squeeze those ventricles. It starts moving the blood, but it's not leaving the chamber as of yet. Remember, the blood starts panicking, starts moving towards those different doors or those different valves. It has to generate enough force to close the valve. Once the valve is closed, now it goes, oh, I can't get out that door. I better run over here and try to go out that door, like the semilunar valves. So there's a period of time when the ventricle's starting to contract, the blood's getting panicked and starting to move inside the ventricle. It's getting panicked. It tries that door, like the atrioventricular valve, and it can't get out that. It just closed that one. Now it has to rush over and exit out the semilunar valve. So at that point, after it's closed that door, and before it's opened that door, technically there's a time the ventricle's contracting, but the blood's not moving anywhere out of the ventricle yet. So it has to first shut that door, then generate enough pressure on that door to open it up. And once the semilunar valve's open, now the blood's being ejected into the pulmonary circuit and into arch of the aorta, and now it's exiting and doing the contraction that we normally think about. But there's this brief period of time when the ventricles are contracting, but the volume isn't changing yet because we have the blood that's actually closing the AV valves and now opening the semilunar valves instead. Okay? Again, it's not something that you usually focus on. You usually are focusing on when the blood is filling and when it's emptying and stuff like that, but there are these transition periods as well. Okay. There's also this one. I'm not going to go through that one. It's kind of the same thing. Isovolumetric relaxation instead. So it is important that we have to generate this pressure so that we can open and close valves and generate this pressure so that we can move the fluid out of those different chambers when necessary. Okay? So we want to make sure that we can open and close these valves as necessary. It has to do with that pressure that's being generated. And once there's enough pressure to move the door, then the blood is actually going to move out of that one chamber. All right. So once the blood moves out of the chamber, now we're getting some output. Okay? This is not chemistry class. This is not carbon monoxide. Okay? CO, when we're referring to the cardiovascular system, is cardiac output. It's our calculation of how well this heart is doing its job in terms of putting blood to the body or putting it to the lungs. Now, generally, cardiac output, like we said, amount of blood pumped by each ventricle in 1 minute. You could say 1 second. You could say 1 hour. Generally, they agree it's 1 minute. Okay? And even though we say out of each ventricle, right, that means it's ejecting out of the heart. A lot of times we focus on the left ventricle more. Why? Where's the left ventricle blood going? To the body. That's where we really see the output being important. It's certainly important to go to and from the lungs, but that left ventricle is really what's serving the body. So when they're doing cardiac output, they're focusing on the ventricles because those are the ones ejecting blood out of the heart, and we're focusing on, a lot of times, the left one more than the right one. Okay, so cardiac output, this is our calculation of cardiac output. It's not very difficult. Cardiac output is equal to heart rate times stroke volume. Now, heart rate, that one's not too difficult to figure out, right? Number of beats per minute. So we decided we're going to look at a minute, so we want to know how many contractions are we getting per minute. That's going to be important in terms of how much blood is spurting out in the horror movie or whatever, right? But also, those individual squeezes that we get, how much blood is being ejected, like we said, when that ventricle contracts, that's also going to be important in terms of how much blood's coming out. So stroke volume is one squeeze. How much do you get in one squeeze? And then how many squeezes are you adding per minute? That gives us total cardiac output, okay? On average, one individual squeeze will be about 70 milliliters of blood. One squeeze. And then, how many of those do you get per minute? Now again, if you're running or if you're excited or something like that, you're going to get more squeezes per minute. You're going to get increased cardiac output. Now, there's a couple different ways that that can increase. Like we said, we can increase the rate, or we can increase the force that we do for one squeeze. Like if you really squeeze the heart and make sure that there is a very low end systolic volume, then you get a lot more blood in an individual squeeze, okay? So when we look at it, like we said, usually an individual squeeze is about 70 milliliters per squeeze, and commonly a resting heart rate might be 75 beats per minute. So if you were to multiply that out, 75 times 70, what you get is slightly over 5,000 milliliters per minute. Now, why does that seem significant there, that volume of blood? Does 5 is a nice round number? That's about how much blood you have in your body in total. So when we were doing the random facts, if you will, having to do with the cardiovascular system, we were talking about the fact that you have anywhere from 4 to 6 liters, on average 5 liters of blood in your body. That's saying that even when you're doing a resting heart rate sitting here right now, every single minute that blood is going around through your heart and being circulated around the system. So it does a whole circuit, if you will, every minute. Okay? Okay. So the heart does a good job of moving this material, moving this connective tissue around the body. So we talked about things that can affect it. We talked about heart rate. We talked about our stroke volume and things like that. But there are some factors that can affect even that individual squeeze. So this says preload, afterload, and contractility. So preload is basically, like we said, when the ventricles are relaxing and they're filling up, how much blood is loading into there. The cardiac muscle has a very interesting property to it, in that the more, to a certain extent, the more stretched it is, the more force it actually generates. When we were talking about skeletal muscle way back when, you probably, it was a while ago, you probably at one time looked at the amount of force that you could generate with an individual skeletal muscle fiber. And if it was overstretched, like if your arm is out like this and this muscle is overstretched, you can't generate a lot of force, right? So if you're at that machine at the gym and you're trying to do the biceps and you have it way out here, you can't generate a lot of force way out there. Meanwhile, if you have that weight and you have it right here and you're doing this type of thing, you're not going to generate a lot of force there either, because the muscle fibers, the thick and the thin filaments, are already overlapped pretty much. So they're not going to be able to generate a lot of force that way. So for skeletal muscle, the optimal length for them to generate the most amount of force is actually resting length. Not overstretched, not already partially contracted. Resting length has the right amount of overlap between thick and thin filaments and generates the most amount of force. So that makes sense. But for cardiac muscle and for smooth muscle, they have the most work to do when they're actually being stretched. When that chamber is filled, whether that's a heart chamber or whether it's the stomach or something like that, that's the time when they need to be working the best, right? If I have a full load of blood in there, or if I have a full load of food in there after lunch, that's when I need strong muscular contractions to move that, right? Not when it's empty, that's not a big deal, when it's full. So cardiac muscle, in fact, when it is stretched more, it actually responds stronger and is able to eject more blood. So preload, the more blood goes into the heart beforehand, the more it ejects. It tries to eject anything it gets back, right? So it's like a karma thing, right? As much as it gives, it gets, or as much as it gets, it gives. So preload, if it is loaded even more, it will eject even more. So that affects stroke volume. Contractility is talking about how much force these individual cells can generate due to some other factors like ion concentration and things like that. Afterload means that when we are pushing blood from, say, the ventricles into those major arteries. What are the two major arteries coming from the ventricles? So we've got our different valves that lead into two major arteries. One that goes to the pulmonary circuit, pulmonary arteries or pulmonary trunk. What's the other big arch one? The aortic, right? So the aorta is another big artery. Those different arteries have blood in them, right? If you were to follow that path downstream, there's blood throughout there. It's not just air there, right? There's a whole blood throughout there. It's a big circuit. So if I push here on this big circuit, all the rest of this has to shift forward like a train, right? So there's a bit of resistance when the heart is trying to push new blood into those arteries from all of the rest of the blood that's throughout the circuit, right? So it's like if you're like lining up to exit a game, like you're leaving another horrible Diamondbacks game where they've lost like 13-1 or something like that. You can't get out of there fast enough, but people behind you are like, I got to get out of here. So they're like shoving you. They can't go faster because there's people in front of them. Same thing in terms of the blood. So when the heart is trying to push more blood into the circuit, it's working against all this other blood that's filling up that whole circuit. So that's called afterload. So it's back pressure that's exerted by the blood that the heart has to overcome. So that's going to affect stroke volume as well. Okay? So this, again, just goes through what I was talking about. This idea of preload, the amount of stretch or amount of filling that occurs will influence how much we put out. And then afterload, like we said, is the resistance that it has to work against if it's pushing new blood into these arteries. So they'll try to exert some forces going back against them instead. Okay, like we said, in terms of other things that can influence cardiac output, different types of ions, different types of hormones, etc., as well as the nervous system stimulation is going to be important in terms of changing that. We can make it squeeze harder. We can make it squeeze a little bit weaker. So the influence of these external factors on the squeeze will affect stroke volume, which then scales up and affects cardiac output as well. Okay? So lots of different extrinsic factors that can affect the heart's activity. This one also is the same thing, just other types of ions. If we block those calcium channels, obviously it's not going to work. If we upset the balance in terms of the concentration, right? If this is a plasma membrane, sodium is supposed to be big outside and only a little bit inside. Potassium is supposed to be big inside, only a little bit outside. If you blast a whole bunch of potassium outside of the cells, then when these channels open up and they're supposed to allow all this potassium to exit, well, if we have a bunch over here, that channel is going to open up and you're not going to get movement. You're not going to get diffusion. So if you don't have diffusion, you're not going to be able to repolarize the heart. One of the ways that you can stop somebody's heart, not like you want to know that type of thing, but one of the ways you can stop somebody's heart is using potassium chloride. So sometimes on those like different medical dramas or something like that, you'll see somebody and they come to the hospital and they inject something into the IV and then that person like flatlines. So potassium chloride is one of the things that can do that because you're loading the external environment with a bunch of potassium. So the heart cells contract, and when they try to relax and repolarize, those channels open up and the potassium doesn't move, so it doesn't repolarize. So it basically stops. Okay. So the ion concentrations are going to be important in terms of what we're talking about in terms of the activity of the heart itself. So what we find is if you have increased or decreased ion concentrations, it can influence the activity of the heart. So remember, hypo means low, hyper means high. Calcium is important, potassium is important as well. So hypocalcemia depresses the heart because we know that the calcium helps to depolarize the heart and maintain it in that state. If you have too much calcium, that makes it a bit spasmodic. It makes it contract more, stronger, etc. Potassium is the opposite, right? So if we look at hyperkalemia too much, then basically we're leading to heart block. And if you have hypokalemia, then what you have is you have different types of arrhythmias, etc. So you don't have enough potassium to generate a full, strong heartbeat itself. Okay? So different types of chemicals will certainly affect the heart. Ion levels that we have to maintain homeostatically, and even hormone levels as well. Now, we'll do endocrine system this semester, but you know this one, right? What's that one? Epinephrine, also known as? EpiPen, sure, yeah. EpiPens, which cost like $600 or something like that right now. Big blow up in the news about that. Price gouging for the EpiPen from this one company, and where the CEO is making millions upon millions. And like, hey, people are going to anaphylactic shock. They need these EpiPens, right? Anaphylactic shock is where your immune system is responding to some sort of allergen, and it doesn't know where it is. So rather than doing inflammation like, I'm going to deliver more white blood cells to this area because it's infected, it's like, I don't know where it is, so we're just going to open up everything. And if we open up everything, the blood pressure that this heart's trying to maintain just drops to the floor, right? It's the same 5 liters, but now it's spread out over a whole lot but larger area. And so the blood pressure and the blood flow basically drops all the way through. So critical organs aren't getting what they need. So you do epinephrine. Epinephrine has an effect, adrenaline has an effect on this system overall. So it has an effect on the heart as well as on the blood vessels. And on the heart, it's going to increase it, right? An adrenaline rush increases heart rate and increases stroke volume, okay? So thyroxine, we'll talk about that one too. That's basically a thyroid hormone. It affects your overall metabolic rate. So this whole section here, we're looking at this, right? Cardiac output. We want to see that the heart is putting out enough blood to supply the body with what it needs. In terms of cardiac output, 2 things go into that. Heart rate, number of beats per minute, and stroke volume, the amount or volume you get in an individual squeeze. And what we've said is that there's a number of things that the body controls or a number of things that can affect those 2 factors that then cardiac output is going to change. Sometimes we're going to change it intentionally, like we said, running on the treadmill or going out running or something like that. We need to increase output. Sometimes our cardiac output is being affected by these things, and it's not intentional, like when potassium levels get off or whatever. Okay? All right. Last little bit. What do we got? 5 minutes? Something like that? Yeah? Keep me on track. Shuffle papers if you need to. I think we got 5 minutes. Last little thing in terms of the heart. Remember when we were going through in lab, we said that the fetal heart and the adult heart is a little bit different because fetal circulation, we have this whole other circuit, right? We have this whole other circuit with the placenta that we don't have in the adult. So in the fetal circulation, we have 4 different circuits. Pulmonary, systemic, cardiac, those are the 3 we all have. And then they have this additional one, which is kind of the umbilical or the placental circuit. So that's where, with the placenta, that's where gas exchange occurs. That's where nutrient and waste exchange occurs. And so it's a little bit different in terms of our heart structure as well as our vascular structure. So we know we have 2 differences in the heart, the foramen ovale and the ductus arteriosus. So the foramen ovale, foramen means what? Hole. And it's an oval shape. So this is a shortcut between the 2 atria because once we've done this exchange down with the placenta, we've got more oxygen and nutrients, etc. We're dumping that into the right atrium. We don't need to send that to the lungs again, right? We already did our oxygen exchange. Let's already shortcut it, send it over to the left side so we can send it out to the body faster. So the foramen ovale allows that connection to occur between the 2 atria. Ductus arteriosus is another shortcut between the pulmonary trunk and the aorta. Again, rather than going all the way to the lungs and back, let's just shortcut it right into the systemic circuit that way instead. Okay? All right, the last few things in terms of diseases, disorders. Like I said, mostly I don't require these for the exam. You should know, you hear about coronary artery disease and bypasses. This is where we're talking about blood vessels that have narrowed or have become blocked, and we need to get around them. It could be due to fatty plaques that have accumulated. It could be because of a clot or a thrombus that has formed in that particular area. So usually they'll try to kind of thin that out. They may try to pack it with a stent and kind of push it along the side and then reinforce it, or they may bypass it altogether. Okay? For arrhythmias, we did mention these when we were talking about the heart itself. You should know the definition of arrhythmia, that is an irregular heartbeat. And then for these ones, these three you should know as well because they are common enough. Whether you've heard of somebody in your family or friends that have had a condition like this, or you'll actually hear it in the medical field, right? Tachycardia is the one that you normally hear of, where the heartbeat is too fast. Bradycardia is the opposite, where the heartbeat is too slow, okay? And then fibrillation is that kind of spasmodic contractions instead. Can occur in the atria, where it's not as serious, more serious when it occurs down in the ventricles, okay? Okay, so with that, it takes us to the end of the cardio part of cardiovascular system. The next thing we talk about, last part, is vascular component instead. So make sure that you have those notes available for next time, and we'll be talking about that next time.